All right, part 2, we're going to be talking about the structure and the parts of the cell. So I'll be bouncing over here on my websites between a couple of different websites. On my left here, you can see an animal cell, and it's got it where you can see pictures from a scanning electron microscope. So you can see rhabdoplasmic reticulum, and you can actually see the ribosomes that are part of that. It's got the nucleus, and you can see the nucleolus inside of it. It's got a lysosome, and you can see its round structure there. That's basically your stomach area, your plasma membrane, and I've got another site when we talk about the membrane I'll show you. Centrioles, which are only found in plant cells, are the anchors for your spindle fibers during cell division. Here's your mitochondria. We always draw it with little zigzaggies, and you can actually see them. That's from a real cell. It's labeled vacuoles. You can't really see those. Here's a Golgi apparatus. See how it's all kinds of bubbles? It packages proteins like in little membranes. And here's smooth endoplasmic reticulum. It looks kind of like the rough, but it doesn't have the ribosomes, ribochondria. And then there's glycocalyx, which is a structure, on the cell membrane. So those are your animal cell structures. Here you've got leucoplasts, which are different storage, like vacuole type things, and they do different jobs in a plant. You've got mitochondria. You've got your cell wall. It looks kind of like this on the outside of the cell membrane. And then the cell membrane's inside that. And below that are chloroplasts. This is a chloroplast, and it's got little discs on the inside of it. Again, your endoplasmic reticulum. Rough or smooth, you guys should be able to recognize it from your last picture. Plasmodesmata, those are your little like bridges in between different plant cells. That's what that line is pointing to is. You've got your nucleus and a very dark, large nucleolus. And you've got your Golgi apparatus again, which is bubbly. And then your large central vacuole, which helps with the water retention of the cell. So we're going to go through your cell. I will actually be looking probably at this site for most of it, just because it's a little easier to see of either a plant cell or an animal cell to point different structures out. So first of all, the plasma membrane or outer membrane of the cell through which oxygen, carbon dioxide, and water pass freely. It is flexible. So actually, for my, hold on, one moment please, what's going on here? I want to look at my most recent apps. There we go. I had a membrane interaction thing up here, and it's a great site. I will share all these sites with you so you can study, but it's got the ability to let you look at different molecules as they pass through and let you see the phospholipids. So that's what I wanted you to see. You've got your phospholipids, which has the phosphate head and the lipid tail. You can throw your big gang signs now if you've got them, so you remember. They form a bilayer, so you've got an inner layer, which the phosphates face the inside of the cell, and an outer layer where the phosphates face the outside of the cell, and then the lipids are in the middle. It is selectively permeable. That means it only allows certain substances to pass through. It's got pores that allow some materials to pass through, but not others. So in this diagram, you can see that there are different pores here. There's the aquaporin, which allows water to pass through. There you can see aquaporin allowing water to pass through. It's got the gluten transfer, which the glucose transfer. It allows glucose to come in, and see how it kind of changes shape as it does that. And we'll get into this in a lot more detail in another place. It's got ion channels that will just, again, they're like the water channels and allow different things to pass through. The sodium and potassium pump, if you notice, two things are happening there. Sodium, it goes out, and potassium comes in. That's important when you do your muscles, and they have to allow things to move out, in and out. So it is inside, it's the outer layer, sorry, letter D. It's the outer layer of animal cells, and it is inside the cell. Only plants have cell walls. Animal cells do not have cell walls. So again, your first blank is plasma membrane, second blank is phospholipid, third is selectively, fourth is pores, fifth is animals, sixth is plants, and only plants have cell walls. I know I didn't have that up while I was showing you that diagram. Number 2, the nucleus. In this diagram, it's this blue structure. You can actually see unwound chromosomes in the nucleus, and you can see the dark spots, which are the nucleolus. It is a control center of your cell, which contains the DNA. This is your genetic information. It's in the form of chromatin right now in this picture, and it will become chromosomes right before the cell divides. It will wind up into chromosomes, but most of the time it's unwound because when it's in its chromosome form, it can't do anything else. Like, it can't be read by the ribosomal, it can't be read and changed into messenger RNA, is what I'm trying to say. So it's surrounded by a nuclear membrane, and those are also phospholipids that surround it, with pores that control the passage of materials into and out of the nucleus. So it's kind of got a place for the material, for that RNA. The chromatin never leaves, but the RNA, after it copies it, the copy of the blueprint can leave, and it will allow that to leave and go into the cytoplasm and do its job. It also contains the nucleolus, where DNA, deoxyribonucleic acid, is concentrated to send ribosomes constructed of RNA and proteins out with instructions for the cell to build proteins. So a ribosome is actually made of RNA and proteins, and the nucleolus is where those are constructed. It controls the cell in response to the environment. In general, cells are regulated by three things: raw materials for a reaction to take place, energy, and the proper catalyst to speed up the reactions. Most reactions are too slow without the catalyst. Enzymes are the catalyst for these reactions. Most reactions in your cells would not occur at all without enzymes, or they would take years and years and years to take place. I have another site. I know you guys are like, just give me the lecture notes, but I do want to show you these things. This is called The Inner Life of Cells. It's by Harvard that made this. I showed it to all my classes repetitively. Where did it go? Oh, my iPad. It is fighting me. So let me see if I can find inner thigh workout hashtag. That's not what I want. Inner life of a cell. Okay, so there's all these pictures, and in here it does show you the nucleus. It does show you RNA coming out of the nucleus and different things like that. So I encourage you to watch it. Oh, it's going to even give me sound. I'm sorry. So about right while I'm trying to talk, but I'll send that link to you, and I encourage you guys to go in and watch it and look at those nuclear pores, examine how the RNA comes out and how the proteins are made. So let's keep going. There's usually only one nucleus per cell. That's your last bit on your nucleus. So there's usually only one per cell. Cytoplasm is everything between the nucleus and the plasma membrane, and includes the many organelles we will discuss. The cytoplasm isn't just jelly, it's got the cytoskeleton involved in there as well. If you watch that video, which I so hope you do, and if you look in here, there's all these like strands of stuff coming in and out and around all the organelles. And that motor protein that was shown in the video was actually walking on one of the microtubules. So stuff doesn't just get randomly shuttered out, nope, that's not the word, transported out into the cytoplasm and hoped for the best that it will arrive at its destination. It actually gets attached to a motor protein which transports it from point A to point B in a very efficient manner. So that's all in the cytoplasm. The mitochondria, of course, is the powerhouse of the cell. If you had AP biology, you know it does oxidative phosphorylation, which means it joins a phosphate group onto ADP to make ATP. Ribosomes build protein in the cell. Those ribosomes are usually found on your endoplasmic reticulum, which is your next note, but sometimes they are found free-floating in cells. The endoplasmic reticulum is a membranous organelle that function as a site for the synthesis of making proteins, lipids, and other materials. So they don't just make proteins, even though that's the main one that we discuss usually in class. The Golgi apparatus will then modify or changes many of the products and prepares to send them out for the cell. In the video, what's actually happening is the motor protein is carrying a package of proteins that were done from the endoplasmic reticulum and need to go to the Golgi apparatus for further modifications. Number 8, your lysosome contains digestive enzymes that break down or destroy bacteria in white blood cells. Now, your lysosomes are usually mostly found in animal cells. They're these red structures here. They're not usually found in plant cells unless your plant is like a Venus flytrap or another type of insectivorous plant. And that doesn't mean that they actually eat the full insects, but they break them down and absorb some of their nutrients because their soil is poor. So if you have a Venus flytrap, for example, in good soil, it's not going to need to eat as many insects. But because of where they evolved, they need to do that. So they would have some lysosomes where most plants would not. So lysosomes, again, contain digestive enzymes that break down or destroy bacteria in white blood cells, or they break down old worn out cells or proteins, etc. The chloroplasts contain chlorophyll, and they are green, and they are only in plant cells, which is halfway true because some protists also have chloroplasts. So I'm going to stop there, and then I will pick up and finish your notes in part 3 today.